The cell cycle is a series of events, including mitosis, that cells go through in order to grow and divide. Cells must go through mitosis to divide because if a cell gets too big, the nutrients will not feed the cell properly. The cell spends most of its life in interphase. This consists of G1, G2, and S phase. S phase is when DNA replicates. The next phase is prophase. In early prophase, chromosomes coil up and become visible. Microtubules, called spindle fibers, form and extend on each side. Meanwhile, astral rays, which are also microtubules, much smaller than spindle fibers, are formed and they float around in the cytoplasm. In late prophase, now that the DNA has replicated during interphase, the pairs of chromosomes are called sister chromatids. The centromere is where the chromatids connect. On the centromere, there's a kinetochores, that is a protein bond, on each centromere. They attach to the spindle fibers, forming chromosomal microtubules. That concludes prophase. The next phase is metaphase. This is when chromosomes line up in the middle on the metaphase plate. The chromosomes then attach to the spindle fibers. And that is the end of metaphase. This next phase is anaphase. This is when the chromosome copies divide. Spindle fibers pull apart daughter chromosomes to opposite sides of the cell. The last and final phase is telophase. This is when spindle fibers disappear and two nuclei are formed. The cell gets ready to split, and this area is called the cleavage furrow. This is the end of mitosis. Last and finally, there is cytokinesis, and this is when the cell splits into two different daughter cells. This is the end of the cell cycle, and now these two daughter cells are ready to divide again.